Antibody engineering. A number of discoveries and technologies have converged to produce a renaissance in antibody therapeutics. Our vision of antibodies as tools for research, useful for the prevention, detection, and treatment of disease, has been revolutionized by these recent advances. This review specifically focuses on what is now called antibody engineering and includes chimeric and humanized antibodies, immunoglobulin fragments, antibody libraries, antibody fusion proteins, and transgenic organisms as bioreactors. As a consequence of refinement in antibody technology, the field of genetically engineered immunoglobulins has matured into an elegant and important drug and reagent development platform. So, antibody proteins that bind to a specific target molecule, antigen, at one specific site, antigenic site, in response to either infection or immunization with a foreign agent. The immune system generates many different antibodies that bind to the foreign molecule. Individual antibodies within this polyclonal antibody pool bind to specific sites on a target molecule known as epitopes. Isolation of an individual antibody within the polyclonal antibody pool would allow biochemical and biological characterization of a highly specific molecular entity targeting only a single epitope. Realization of the therapeutic potential of such specificity launched research into the development of methods to isolate and consciously generate a supply of a single lineage of antibody, a monoclonal antibody, which we shortly represent as MAB. In 1974, W. Koehler and M. Milstein developed a process for the generation of monoclonal antibodies. In their process, fusion of an individual B cell or B lymphocyte, which produces an antibody with a single specificity but has a finite lifespan, with a myeloma, basal tumor cell, which can be grown indefinitely in culture, results in a hybridoma cell. This hybridoma retains desirable characteristics of both parent cells, include in producing an antibody of a single specificity that can grow in culture indefinitely. So, generation of monoclonal antibodies through the hybridoma process worked well with B cells from rodents, but not with B cells from humans. Consequently, the majority of the first monoclonal antibodies were from mice. It administered into humans as therapeutic agents in experimental tests. The human immune system recognized the mouse monoclonal antibodies as foreign agents, causing an immune response which was sometimes severe. Although encouraging improvements in diseases were sometimes seen, this response made murine mouse antibodies unacceptable for use in humans with a functional immune system. Fueled by advances in molecular biology and genetic engineering in the late 1980s, efforts to engineer new generations of monoclonal antibodies with reduced human immunogenicity have came to fruition. So today there are a number of clonal antibodies approved for human therapeutic use in the United States. Characterization of the structure of antibodies and their genes laid the foundation for antibody engineering. In most mammals, each antibody is composed of two different polypeptides, the immunoglobulin heavy chain and the immunoglobulin light chain. Comparison of the protein sequences of either heavy or light antibody chain reveals a portion that typically varies from one antibody to the next, the variable region, and a portion that is conserved, the constant region. A heavy and light chain are folded together in an antibody to align their respective variable and constant regions. The unique shape of the co-folded heavy and light chain variable domains creates the variable domain of the antibody, which fits around the shape of the target epitope and confers the binding specificity of the antibody. So, mice genetically engineered to produce fully human antibodies allow the use of established hybridoma technology to generate fully human antibodies directly without the need for additional engineering. These transgenic mice contain a large portion of human DNA encoding the antibody heavy and light chains. Inactivation of the mouse own heavy and light chain genes forces the mouse to use the human genes to make antibodies. Current versions of these mice generate diverse polyclonal antibody response, thereby enabling the generation and recovery of optimal monoclonal antibodies using hybridoma technology. Disease areas that currently are especially amenable to antibody-based treatments including cancer, immune dysregulation and infection. Depending upon the disease and the biology of the target, therapeutic monoclonal antibodies can have different mechanisms of action. A therapeutic monoclonal antibody may bind and neutralize the normal function of a target. For example, a monoclonal antibody that blocks the activity of the protein needed for the survival of a cancer cell, causes the cell's death. Another therapeutic monoclonal antibody may bind and activate the normal function of a target. For example, monoclonal antibody can bind to a protein on a cell and trigger an apoptotic signal. Finally, if a monoclonal antibody binds to a target expressed only on diseased tissue, conjugation of a toxic payload, effective agent such as an chemotherapeutic or radioactive agent to the monoclonal antibody can create a guided missile for specific delivery of the toxic payload to the diseased tissue, reducing harm to the healthy tissue. See antibodies, antigen, genetic engineering, immunology. Applications of antibody engineering. Antibody engineering is used in the treatment of numerous diseases and many diagnostic tests. Each antibody binds very specifically to only one type of cell or molecule, and antibody technology allows researchers to target specific cells. The applications of antibody engineering are successful due to the specific binding of antibodies and antigens. Production methods of antibodies continue to progress, making it possible to generate antibodies specific to almost any antigen. Emerging applications of antibody engineering include cancer therapy and radioimmunodetection of tumors. Antibodies present many exciting possibilities to the future of medicine. Antibody binding specificity. Antibody engineering has become an emerging field because of antibodies' ability for specific binding. The reason antibodies have such specific binding properties is because of their structure. Antibodies are composed of two identical heavy chains and two identical light chains. The light chains make the arms of Y-shaped antibody which have identical antigen binding sites at the end of each arm. The heavy chain makes up the tail of the Y-shaped antibody which contains a binding site for molecules that activate other parts of the immune system. Antibodies are created in the body during an immune response. During an immune response, antigens bind to B lymphocyte cells in the body. There are millions of different kinds of B cells in each body, each specific for a type of antigen that may be encountered in a lifetime. They lie dormant until an antigen binds to them. Once an antigen binds to its specific B cell, the B cell differentiates into two types of cells, plasma cells and memory cells. Plasma cells are antibody producing cells that secrete large amount of soluble antibodies that are able to bind to the specific antigen, and memory cells are cells that live for a long time that are able to respond quickly if they encounter the same antigen as before. The ability for the antibody to recognize a specific antigen it is targeted for allows antibodies to be very specific when targeting antigens. Then production of antibodies that target a specific antigen. Production of antibodies that target a specific antigen are found rather than designed. The gene for the antibody of interest is narrowed down from an antibody repertoire library containing 10 raised to 5 to 10 raised to 10 distinct immunoglobulin genes. There are three types of antibody repertoire libraries: immune libraries, naive libraries, and synthetic libraries. Okay? Immune libraries. Challenging the immune system of mammals, especially mice, with a particular antigen and collecting B cells forms immune libraries. These B cells immunoglobulin genes have already undergone affinity maturation. This means that the immune response initiated in the mammal causes a very high percentage of B cells to express genes for antibodies that target a specific antigen. Immune libraries can be fairly small because most of the genes they contain are relevant to this one, like antigen. So antibodies produced as, however, the antibodies produced are made by animal immune systems and trigger an unfavorable immune response in humans because it is unethical to immunize humans with antigens. Yeah, and that is something which I don't think people will, accept. But I think that is a way of doing research, and only we know how the system works. Have been developed to humanize the genes in immune libraries, but this is not ideal. Immune libraries are also not effective at targeting self-antigens. Naive libraries. Naive libraries do not require immunization and are the food derived from human B cells. Unlike immune libraries, these libraries are very large and have wide variety of antibodies represented. With such a large selection, an antibody to almost any antigen can be derived. This also means a naive library can be used repeatedly to select a different kind of antibody every time. Unfortunately, the lack of immunization means that immunoglobulin genes have not undergone affinity maturation, resulting in antibodies with lower antigen affinity. With effort, this can be partially compensated for by increasing the size of library. Then synthetic libraries. This final class of libraries relies less on organism's immune system and more on creative biochemical engineering, randomly cloning and combining different immunoglobulin immunoglobulin gene segments creates genes that can produce a wide variety of antibodies. These fully synthetic libraries generally do not produce antibodies with high antigen affinity. However, so a better portion is a semi-synthetic library. Semi-synthetic libraries are made by taking existing antibody genes and using PCR to randomize key areas called hypervariable regions. This creates antibodies with specificities for many different antigens. Because these libraries are made without much interaction from the human immune system, they produce better self-targeting antibodies necessary for targeting cancer cells. After producing an antibody library, there are two methods for sorting the genetic information and producing antibodies in large quantities. The first method uses hybridomas to store every individual B cell, while the second method uses bacteriophages to store only the immunoglobulin genes. Hybridomas were first created in 1975. They consist of animal B cells fused with myeloma. Sorry for the disturbance. Hybridomas were first created in 1975. They consist of animal B cells fused with myeloma cancer cells. The B cell provides the immunoglobulin gene and the myeloma provides the ability to infinitely duplicate. Hybridomas are meant to differentiate and secrete antibodies which can then be used for practical applications. Unfortunately, they can only be made from animal B cells, so while they can store immune libraries, they cannot store synthetic or naive libraries. So antibodies made from hybridomas will therefore produce an immune response in humans if they are unaltered. Hybridomas are also problematic because they cannot be made from plasma immune memory cells and a great deal of time is required to isolate a hybridoma for a specific antibody. Although technology is beginning, one scary thing is nothing else. Although technology is beginning to move beyond hybridomas, they have been crucial to the advancement of monoclonal antibodies. Then phage display libraries. Phage display libraries are a collection of immunoglobulin genes rather than entire B cells. So unlike hybridomas, they can store genes for immune libraries, naive libraries or synthetic libraries. Immune and naive libraries are derived from B cells, so the genes are transferred to bacteriophages in several steps. First, mRNA from the B cells undergoes PCR with reverse transcriptase to form a complementary or cDNA library. The cDNA is then amplified and cloned into bacteriophages so that each phage contains one immunoglobulin gene. The reverse transcription is not necessary for synthetic libraries because they are already expressed as individual genes. Phage. Phage display libraries can be refrigerated for storage and expanded by infecting bacteria. The phage DNA is engineered so that each phage has the antibody protein it encodes expressed on the outside of its protein coat. This property allows a specific immunoglobulin gene to be isolated from a phage display library far faster than from hybridomas. Isolate a gene in a copy of a phage display library. The bacteriophages are put in an environment where the targeted antigens are bound to a surface, cell or particle. The phages displaying the correct antibody bind to the antigens and the rest of the library can be easily removed, leaving behind only the desired phages. A few or more rounds of panning narrow the antibody down completely. When a final candidate has been selected, the phage undergoes analytical tests such as ELISA or enzyme-linked immunosorbent assay phage. So, infecting a bacterial colony with the phage produces antibodies on the outside of phage particles. This form can be useful. It is also possible to engineer the phage to infect bacteria and produce antibodies that are independent of the phage protein. This does not produce the entire antibody, just the regions most necessary for antigen binding. Which regions are selected and how they are linked together varies depending on what the antibody will be used for. Cancer therapy, previous method, chemotherapy. New method, antibodies. Monoclonal antibodies have been discovered recently to be useful for cancer therapy, with some treatments gaining FDA approval. It has been determined that some antibodies bind specifically to tumor associated antigens, which are present in higher concentration on tumor cells than normal cells. The antibodies then begin to accumulate in malignant tumor tissues, which can then recruit cytotoxic immune cells or can be combined with a drug delivery mechanism to deliver chemotherapy drugs. A monoclonal antibody has been used for the treatment of large cell lymphoma in conjunction with chemotherapy. Studies show that after 1 year of treatment, 69% of patients treated with this rituximab drug in conjunction with chemotherapy were free of lymphoma, versus only 49% of patients treated solely with chemotherapy were free of lymphoma. Monoclonal antibodies. The use of monoclonal antibodies for cancer treatment is very promising. Then cancer targeting monoclonal antibodies. One type of monoclonal antibody that has been FDA approved is Avastin or bevacizumab, which is used to treat metastatic colorectal cancer, non-small cell lung cancer, and metastatic breast cancer. Avastin, reach beyond convention. Bevacizumab is a recombinant humanized monoclonal IgG1 antibody that binds to the vascular endothelial growth factor and inhibits its activity. This drug is able to target VEGF specifically because it contains human framework regions and complementarity determining regions of a murine antibody that specifically binds to vascular endothelial growth factor. The bevacizumab antibodies prevents VEGF from interacting with its receptors on the surface of endothelial cells, which cause the endothelial cells to proliferate and new blood vessels to form. In mice with xenotransplant models of colon cancer, the drug reduced microvascular growth and slowed progression of the metastatic cancer. It has been found that the treatment of cancer is best paired with Avastin and chemotherapy for best results. For intravenous use, although monoclonal antibodies have shown promising results in the treatment of cancer, dangerous side effects can occur. In Avastin, some dangerous side effects that have been seen are gastrointestinal perforation, wound healing applications, and hemorrhage. In clinical trials of Avastin, all three dangerous side effects resulted in fatality. Antibodies may be used as a more effective cancer treatment than chemotherapy in the future. Since monoclonal antibodies have the ability to target cells directly that exhibit the designated antigen, this treatment may one day be used instead of chemotherapy, which affects all cells in the body. Since there are still dangerous side effects of using monoclonal antibodies as treatment, an antibody would have to be developed that don't cause complications. Then imaging technology, new technology, radioimmunodetection. Monoclonal antibodies are also used for the radioimmunodetection of tumors. Although this technology, antibodies are tagged with radioisotopes whose presence in tissue can be detected with medical imaging scanners. Goldenberg. Once the antibodies have been injected into the body, they target tumor cell surface marker antigens. The resulting image can detect small tumors and can tell more about the stage of the cancer. Using antibodies to detect the most common tumor cell surface marker, the carcinoma embryonic antigen, has been shown to detect tumors as small as 0.15 cm. The use of radioimmunodetection could result in earlier detection of small tumors in early treatable stages. Other uses of antibodies. Drug targeting monoclonal antibodies. Monoclonal antibodies have become very important in engineering targeting drugs because cell surface have many receptors that can be recognized by engineered antibodies. When developing a monoclonal antibody to be used as a medication, there are several factors to take into consideration to develop successful drug. Most importantly, the antigens that the monoclonal antibodies recognize should be localized on the surface of a targeted cell. This would cause the antibodies to have tumor tissue specificity. Also, the antibody should be localized at the target site, and the toxicity of the engineered antibody must be taken into consideration. The drug complex then should be biodegradable and non-immunogenic, as not to trigger an immune response upon treatment. Ideally, the drug should be released when the target cell and carrier molecule interact. Virus targeting monoclonal antibodies. Recently, scientists have been working to develop virus targeting monoclonal antibodies. There have been promising studies in monoclonal antibodies targeting virus. It was found that an engineered monoclonal antibody was able to cure West Nile virus in infected mice. This experiment was the first successful demonstration of the use of a humanized antibody as opposed to exposure therapy against a viral disease. In the experiment, when the engineered monoclonal antibody was used as a treatment in mice infected with West Nile virus, over 90% of the mice survived. Normally, without any treatment, the mice only had a 10% survival rate. This antibody was successful because it either bound to the West Nile virus envelope protein or it bound to the virus in a way that virus was neutralized. From these successful trials in mice, scientists have gone on to create a monoclonal antibody to treat West Nile virus in humans. This drug has been approved by the FDA for clinical investigation. The humanized monoclonal antibody has been shown to neutralize the virus in vivo. Most monoclonal antibodies engineered to target virus infected cells are still in their clinical stages. Even so, there have been other monoclonal antibodies that have been engineered to target cells infected with HIV, respiratory syncytial virus, swine vesicular disease virus, and many others. Respiratory syncytial virus infections, a monoclonal antibody drug palivizumab was been engineered to treat this virus. Palivizumab is a humanized monoclonal antibody targeting an epitope on the virus antigenic site of the F protein for intramuscular administration. Virus targeting monoclonal antibodies may be able to become a treatment for dangerous virus in the future. The recent experimental work could lead to a more effective treatment for HIV. Virus targeting antibodies is just one application that the future of antibody engineering may hold.